Swayam Prabha. Digital India. Educated India.
Shall we start? Okay, all right. Okay, so thanks for the introduction. As he already said, I am working in the Department of Biotechnology. And I'm trying to look inside a cell, like how a cell looks like. Do you all know what a cell is? You should know that, right? Yeah, yeah. So have you all seen cells, like under the microscope or this? So which cells have you all seen? Okay. Have you seen cheek cells? Yeah. Okay. So that would be something that you would have seen. Okay. Can you see a cell by your eye? Egg you can see, but you can't see your body cells and all, right? Do you know why? It's too tiny. So, is it in nanometers? Is a cell in nanometers? Yeah, micrometer. So you all know what a micrometer is? Minus 6. Minus 6. What is millimeter? What is 10 to the power minus 9? Very bright kids. Okay. So, so this is what you would have seen. This is like a blood smear. So if you just poke your hand and is RBC, these are WBCs. Have you all seen this ever? Yes. In a diagram you would have seen, but you may not have seen it like this, right? Sorry? Yeah. So you would have seen it in the slide. Have you seen this? Have you put curd, yogurt you eat? If you put that on some kind of a slide, lactobacillus. So you can see lactobacillus as well. So these are all in the order of RBC is about 6 to 8 microns or micrometers in diameter. Bacilli and all are about 1-2 microns. So when you see a figure like this, if you see in scientific publications and all, you will have this line. This is called a scale bar. Okay, so this is important to understand the size that you're looking at. See, I can show you different images. I can show you this image and this image. They both are at the same size. But the magnification that you're seeing or the details that you're seeing are at a different size within the image, right? So we always give a scale bar. So in this case, the scale bar is about 10 micrometers. Okay, so that gives you an indication that this much distance is 10 microns. So if you're seeing a small thing, bacteria, you can roughly divide this into like 5 and then 1 to identify what is the size of the object that you're looking for. So whenever you see an image in any textbook or in any paper, you should always see the scale bar, whether it is nanometers, whether it is micrometers, whether it is millimeter. So you get an idea of the size. Now tell me, have you seen inside a cell in the microscope? None of you all have seen. Have you seen a virus? Yeah. So have you seen it in the microscope? Have you seen this? What is this? So how did they get this picture? So you already know this. Okay. So electron microscopy is one of the ways to see viruses or within the cell. Now, I'll try to take you to why we can't see it by a light microscope, but we need an electron microscope to see this, and what you can do with an electron microscope. That is also some of the research work that I do, and how we can make like these, this is a projected image, but it's actually a 3D volume. You can make a whole 3D volume using an electron microscope, okay? So one thing that you all clearly explained is that there is a difference in the size, right? So you have things in millimeter range, you have micron range, then you have nanometers, and you go below nanometers. So you go to angstroms. Angstroms is 10 to the power minus 10. So what we were looking at, like RBCs and all, are in the range of 1 to 10 micron. That you can see by light microscopy. You can understand how they are assembled, how they are organized and stuff like that. But if you want to go below that, if you want to look at viruses, say in the range of nanometers, or you want to look at what is inside the cell, you know, all these different organelles like nucleus, mitochondria, ribosomes, all that if you want to look at, if you want to go below that, you need to go into the range of nanometers. And there is a physical limit to how much you can see using light. So all these regions you cannot see by light. And why that is, I will try to explain. Yeah. So first of all, there are a few concepts of imaging. So when you are looking at something, there are a few concepts that you need to understand. One of them is magnification. So you all understand magnification, right? It's the apparent size in the image that you see of the object. You are not actually increasing the size of the object. It is just an apparent increase in the size that you see because of the optics of the microscope, right? So very easily you can define magnification as size of your image divided by size of your object. So if you have like a tree, you can take the height of the image, you divide it by the height of the object. That will give you how many times you have magnified the object. So normally when you're looking in a light microscope, what you would have seen an onion cell, you will generally have like a 4 or 10x eyepiece, and then you will have a 60 or 100x magnification objective. So if you combine these two, you can increase the magnification. So if you have an eyepiece of 10x and if you have an objective of 40x, you can do a 400 times magnification. So you can multiply that. So that way you can see many details of a cell. For example, if you have like a root tip, at 50x you'll see some details, then at 100x you'll see other details, and then you can go on like this up to 1000x where you can start seeing more details. Like that for human blood cells also. Like at lower magnification you see the overall structure, and as you go on increasing the magnification you start to see the details. So by this principle, I should be able to combine lenses and get higher magnification, right? Like with light also I should be able to do it. But that doesn't happen. Does anybody know why? Yes, so that's the exact answer actually. So the reason why theoretically you can combine 100 lenses and get a magnification of 1000 or more than that, right? But it doesn't work because there is a physical limit. So other than magnification, there is one more parameter which is important, which is said is the resolution. So you would have seen, like if you have an image, you can't keep zooming into it, right? Like at some point it becomes blurred, that you can't see the details of that. That is what we call as the resolution. So resolution you can roughly define as the distance between 2 points that you can very clearly see. So if you have, is there a chalk? So if you have an image, right? So this is made up of many small dots which make up the image. So each of these dots, how well you can separate out, that defines the resolution of your image. So this distance, like how well you can distinguish the 2 dots in an image, will tell you how well it is resolved. So if you can resolve only this and this, it's a very long distance. So the resolution of the image is also poor. Whereas if you can distinguish these small details, then the resolution of the image becomes better. So if you look at this in a cross section, like if you have a dot, so if you have 2 dots, if I'm drawing a cross section through this, you can see it as 2 peaks, like if I'm just plotting intensity, right? So these 2 points, if you can very clearly distinguish, for example like this, then you can say those two spots are well resolved. They are nicely separated, so you can resolve them. In this case, you can't make out if this is one or two spots. They are very close together. So we say it's not resolved. So it's unresolved there. Whereas the third case is at the borderline. That's the criteria. We call it the Rayleigh criterion, where you can make out that it is two spots. It's not very good, but you can make out that it is at least two spots over there. And that is a minimum distance you need. So when you have the maxima of one wave coinciding with at least the minima of the other wave, we call it the Rayleigh criterion. So that is how much you can say is the resolution of your image. Okay, so that gives you a number of how well your image is resolved. Now this resolution, as he said, depends on the wavelength of the illuminating source you are using. So now you can use it by this equation where the resolution is 0.612 into lambda divided by n sine alpha. So this is so this is derived from optical formula. So you get it, I think, from many optics that they would have. You would have seen 1 by 2. You would have seen. So you can have different values of this. Based on the different optical formulas, it'll be roughly about the same. So it'll be 1 by lambda by 2 n sine theta. So that will be about 0.5 lambda sine theta. So roughly similar range, but this one is a bit more accurate value, so we use this one. So 0.612 lambda. So you all already know this. Lambda is the wavelength of light. And this whole quantity we call the numerical aperture. And n is the refractive index. You all know what a refractive index is? You all know refraction also, right? Yeah. So n is the refractive index. And sine theta or sine alpha. So if you have the objective and you have your sample, the objective makes an angle with your sample. So half of this, which is called the aperture angle, that is they've used alpha here. I think you can use alpha or theta. So sine of this whole, this is the aperture angle. So all of this gives you the numerical aperture, and this is this is what the resolution of your image depends on. So if you have light, for example, you know that light is made up of spectrum. So you have violet to red. So if you have 400 nanometers to 700 nanometers, that's the wavelength. So if you plug this in, so I'm plugging the lowest value here. So if I put 0.612 into 400, and then I'll tell you that the numerical aperture of usually a light microscope is 1.4, you can actually calculate it to be 175 nanometers, right? So that is 175 to 200 is the limit of what you can image using a light microscope. So 175 is when you have a perfect microscope, but no microscope is perfect. There will always be some defects in the way the lenses are made, or there'll always be aberrations in your lenses. So you'll always have it slightly lower than that. It never even reaches 175. Now remember, I showed you the scale before, right? So we can image up to 175 nanometers. So up to here, we can image with a light microscope. So you can use all of this. So tissues, cells, and all you can see with a light microscope. But below that, if you want to see, you cannot use light itself. So what do? What can we do? Yeah, so we can change the source of your illumination itself. Seen this. So this is lower wavelength and this is higher wavelength, higher energy to lower wavelength. So this is a spectrum of all the kind of electromagnetic radiations that you have. And generally visible light is over here. So you can use theoretically any of these for imaging. You all might have heard of X-ray diffraction, like which is used X-ray crystallography. You can use X-rays as well to make or look into the structure of a cell or the proteins within a cell. That is one way you can do. Today I won't be talking about X-ray diffraction. Today I'll speak about electron microscopy. So one way is to use that, and the other is what we now know, that is the electron microscope, where you use subatomic particles, and depending on how you produce these electrons, you can have them have different wavelengths for them, but they are in the order of picometers. So this is 10 to the power minus 12. So the theoretical resolution that is possible with an electron microscope is very high. So you can actually go to that level, but usually we don't get information to that level because these radiations are also damaging. So it damages the structure of the cell that you see. Okay, so electron is your source. Now, just wanted to tell you briefly about how it came about, like how electron microscopy came about. So in about 1928, which is like very long time ago, Ernst Ruska, he was trying to build an oscilloscope to measure these electrical pulses. So he was trying to focus electron beams. And that is when he ended up making an electron microscope. You all know this concept of serendipity? Yeah. So accidentally you will have a discovery. Do you know how penicillin was discovered? Yes. So he left the plate, Alexander Fleming, he left the plate, and he found that there were regions where there was no bacterial growth. And that's how he found out that there is some compound which is being produced which can be used to inhibit the growth of bacteria. He was not looking for it. He was doing some other experiment and accidentally found this. So he was also trying to do something else. Accidentally, he ended up focusing electron beams, which is what is needed in an electron microscope. Any microscope, you want to form an image by focusing of your electron, of your illuminating source, right? So that's what he ended up making. Initially, when he made the microscope, it had a magnification about 17x or so, which is very small, which I showed you. You can get in your light microscope also that level of magnification, right? But later on, he combined with a company, and then they were able to get much higher magnification, like in the order of 10 to 20,000. So after that, his brother actually took these images. So these were kind of the first images of pox viruses. And down you can see they write the magnification. This is like an old style of writing the paper. So there, here also they have shown the scale bar. So scale bar is very important when you make your images and all. So this is about 1 micron. So you can see the magnification is about 23,000:1.
Can I ask how focusing electron beams, how does that create a magnification?
So it's not focusing that creates a magnification, because you are able to reach the wavelength, reach higher wavelengths. You can pass electron beams through lenses, which will create a magnified image. So with your lenses, you can create a magnification.
So how does that exactly work?
Yeah, so I will show it to you when I show you the diagram of an electron microscope, just like how we do it in lenses, how we do, I'll compare light and electron microscopy. So that time I'll show you how. So in an electron microscope, the features are a little different. They are not exactly the same. So we don't have glass lenses, but I'll show you what we do instead of that, okay? I'll come to that, yeah? If I forget, just remind me again, okay? So, yeah, so you get a very high magnification, about 23,000:1. This is actually, in today's time, less. So today's microscope, we have one on campus actually. It's actually just behind this building. And it's about 6 foot tall. What you would have seen is a light microscope, right? Which is kept on your table, mounted on a table. So this is like a 6 foot tall instrument. And you have to be careful while operating it. You need to be slightly specialized user for that. So this is how it looks. It's inside a cupboard. So if you just see it, it will just look like a cupboard. But inside you have this whole complicated assembly. The principle is the same. You have an illuminating source. Your sample is there. and then you have a place to detect it, like your image, okay? I'll go into more details of this to tell you all how it is different when you're using a light versus electron microscope. But what I want to show you is magnification wise, you can now go up to 100,000 to 200,000x. So it is very high magnification, and very routinely you can get 2 to 4 angstroms of resolution. So I'll show you some structures as well that we've gotten of viruses, of inside the cell and all, how you can very easily image this nowadays, yeah. And in 2017, this technique, cryo-electron microscopy, got the Nobel Prize in chemistry for, it is actually pushing the boundaries of how to look inside a cell, how to look at the structure of proteins. So that's that. So, okay. So now coming to a little bit of details of how you operate, or how does the electron microscope look like. So first of all, to understand, you would have all seen a light microscope, okay? So this is an upright microscope. So you have like an illuminating source, so your light is from below, and then you put your sample over here. So your sample will be generally on a slide. You will put your blood or your cheek cell, your onion cell, whatever you're doing, smear it onto a cell, onto a slide, and then you will put that on the stage over here. Here is your objective lens. So this is what is contributing to the magnification. And then here is your eyepiece. So your eye is your detector, basically. So you can see the image that is being formed. You can, in some cases, transfer it to a computer and see it on the computer. But most likely in the light microscope, you can see what image you're seeing. So your eye can detect it, right? So very simply, the diagram looks like this. So you have a light source, you have a condenser lens which focuses the beam, you have your specimen, and then you have your objective here, and then this is your detector, which is your eye, okay? Now in an electron microscope, what I'm going to do is, it is overturned. Everything is turned upside down. So I'm going to turn this microscope upside down, okay? And then change everything in an electron microscope. So your source of electrons, or your beam of electrons, comes from the top, okay? It's on the top that it produces an electrons. And then you have your specimen, which is put on a very tiny support. It's not a slide. You don't have a slide over here. A very tiny support where you put in your sample. I actually have gotten that support. I'll show you all. It's a very tiny one, which you put inside the electron microscope. And then you have lenses here, which will help you, which act as the objective lenses. And finally, you have the detector. And this detector is not your eye. You don't see these by eye. You transfer it to a computer, and you see these images on a computer. So whatever you are imaging in an electron microscope, you will see on an associated computer. You don't see it with your eyes, okay? So this is how it will look like. Generally, there are different kinds of microscope. This is one of the kinds that you'll see in many departments. So here on the top, you have an electron beam source. Then here you have your sample entry, and then you have an objective, and then you can see the image. Yes. Yeah, so that is a difference. So this is transmission. So you have two kinds of electron microscopy that you can do. So as he said, there are two kinds of electron microscopy that you can do. One is called scanning electron microscopy. And the other is transmission. So we call it SEM and TEM. So the setups are a little different for both of this. Scanning electron microscope, you have an electron beam which scans the surface of your sample. So if you have, for example, on a surface you have some cells, you have an electron beam which will scan this, and then here you have a detector which will tell you how the surface looks like. So it gives the features of the surface of your. So if you're looking at bacteria, you will, if you just check out on the internet, you'll see some nice images of bacteria and all which look like, you can see the three-dimensional structure of them. What I'm showing here is transmission electron microscope, in which where your sample is, the electron beam is going through the sample. So you are collecting it over here. There is magnification because of interaction. And then here in a detector, you will see your amplified image. So there are two kinds of microscopy. So what you are seeing here, this is a transmission electron microscopy. So this gives you information about the structure of the cell and how the proteins look inside. All that you can get from a transmission electron microscope. Yeah. Okay, so I wanted to tell you also how this happens. Basically, the general principle remains the same. So you have electron source, you have lenses which will make your electron beam parallel, condenser lenses. You have your sample of interest, then there is an objective lens. So this kind of magnification happens because of interference between the electron, but I'll just explain to you all, okay? And then you have here down in the detector. So I'll just go through very briefly. I wasn't planning to explain a lot of details about electron microscope, but we'll just give you details of how the whole setup is made and how the process is. So electron beam, so first is the source. Now light you can produce by a torch, right? If you want an electron beam, electrons are subatomic particles, right? So you have to release it from something. Like all of us have atoms which have electrons around it. So to release it, you can provide energy. So that is called the work function. So any metal you can heat up, and beyond its particular work function, it will release electrons. And that you can use as an electron beam. Okay? So there are two ways of making it release electrons. One is you can heat it, which we call thermionic emission. And the other way is you can apply an electrical field. By that also you can pull out the electrons. So this way you can make an electron beam. Okay, that is your source. Then electron beam is one beam of electrons which you can use for imaging. Okay. The second part that you need to know is lenses. So electrons are subatomic particles, right? So they don't interact with glasses and all. So you can't use glass. So what we use here is a coil. So we use a metal coil to deflect this electron beam. So coil, you all know about, if you pass current through a coil, there's an electric field that is produced, right? So, sorry? Yeah, so if you pass current through a coil, there's an electric field that is produced, and that can actually be used to deflect the electrons, right? So you can make coils which act as lenses. What you want to do in a microscope is you have your source of light, you have an image, there is interaction between the particles or the wave. So you know that light has both dual nature. So light behaves as a wave as well as a particle. There's interaction between the waves, and what you see is the image. Now you want that interaction to happen and you want to focus that. That can be brought about. So maybe I'll show you all the diagram actually, which will, so that you'll understand. You would have seen this diagram, but what happens is something that is not oftentimes clear, right? So normally you would have seen this diagram. You have one, this is your object, and then you have a lens. So I have interaction, and then it will go at the focus, and then through the center of the lens, it will go like this. I've not drawn it to scale properly, but you will see a magnification here, right? So this point we'll call the focus. So what happens actually, what you were saying, how do you get a magnification? So here at this stage, this we call the back focal plane. So have you all thought why we get this magnification as she was saying? So what happens is there is interaction of the light with your object, okay? And light is composed of different waves. So they will all interact with the object in a different way. And that then interferes. You all know about this Young's interference experiments, single slit, double slit experiment. So you know that there is kind of, so how does that come about?
Basically, now there's a split, and then you pass electrons through it, then the chances of it's like a bell curve like probability now, but then with light you can tell that it doesn't technically, it's not technically like restricted to the slits, it goes to different cases, but then the probability of it being one part is higher than the other.
Okay. Yeah, so there is a diffraction pattern that happens. You all are clear about that, right? So if you have a slit, so this is a slit, there is, so when light passes through this, you will have like waves formation. And there will be interference. So there is interference in these regions. So there will be both constructive and destructive interference. So when there is constructive interference, you will see a bright line of light, and where there is destructive, you will see no light. So there is an interference between the waves. So you all know about constructive and destructive interference, right? So now when image formation happens, that also happens. So an image is made up of different atoms, right? So there is interaction of light with these different atoms in an object, and they'll all interact differently. So when I have an object, I have a light source, and then there is interaction, and then the light comes out. So they are all now in different phase, different amplitude, everything. So this will interact with each other and will cause a magnification. So that is what you see as your object. But you have to collect all of this rays. So all of this collection is done using a lens. So you have a lens to bring all of them together, and that will cause a magnification. Yeah, is that fine? So in electron microscope, instead of actual lens, glass lens, you have a coil which does the same job. It will bring in all the electron particles together, and that will give you a magnification that you want to see. Yeah? Okay, so when light interacts with the object, there is so there are different interactions that happen. You can have light passing through the object. You can have elastic scattering. So that means it is just going to change the path, but not really change the energy of the wave. And there is inelastic scattering, where even the energy of your wave is changed. So the image is formed because of elastic scattering and unscattered. So there is interference between that, and you see an image that is being formed. It's the way there is the light interacts with the object that you will see this kind of an image. Yeah.
So the lens like collects all the interference and projects the image.
Yes, and that's where you get a magnification. So from this step to this step, then you get a magnification because of that. So that's what happens in electron microscope as well. So the electrons will interact differently with the atoms in your object, and there is interference between the different interactions that happen. And after that, your coils will collect all this, and then you get a magnification. Now, because in electrons, the wavelength is low, you can theoretically go to very, very high resolutions. So that is the way electron microscopy is done. So apart from lenses, even the support that you use, like to make your sample, that is also different in electron microscope. So you will use a glass slide, right? In your light microscope, you all have seen a glass slide. So in electron microscope, we have a very small electron grid. I've actually got a grid which I wanted to show you all. So this is a grid. I'll pass it on. This is your slide that you use for an electron microscope. It's like your bottu, you know, like that's the size of it, on which you put your sample. So your sample can be a cell, it can be a purified protein. I don't know if somebody has spoken about protein purification. You can also get protein out from the cell and you can put it onto this support. So I'll pass this on. There are two of them. You can just see that's how an electron grid looks like. And this small thing is where you put your sample and then you mount it onto an electron microscope to see it. Now that I'm going to pass it on to you all, even that small thing, actually inside it there are squares. It's a metal, it is fabricated, it's a metal, small metal grid on which there are squares. Inside that square there are holes where your sample is suspended actually. So that is the level that you are looking at even for sample preparation. So you will just need about microliters of sample or very, very little sample that you put on it, and that can be mounted on the microscope. So when you look at the side, so inside this there are actually holes. You can't see it like this. I need a microscope to show you what looks inside it. Like I need a light microscope to show you how the grid looks like. So inside that there are holes where you will have your sample. So it will look like this. So this is your metal grid support. In between that your sample is hanging, like because of surface tension there will be sample over there. And so then electron beam interacts with this sample and you get the image below. That's how you see it, right? And I'm not talking more about the detector. We also have a slightly complex detector, like the electron information needs to be changed to charge, which is then you see it on a computer. So it's similar to CCD cameras where you detect a photon and then that's changed into a charge. Here you detect an electron and then that's changed into a charge, which you can then see. So it's generally a detection that happens on a screen that you see in your electron microscope. Okay, so this is very basic. I can, if you all are interested, I can tell you all places where you can read more about it, right? Yeah. So what can you do with this, right? So I just wanted to show you all one basic simulation how when you are, so there are two in that actually, the grids. One of them, you can move it around a little bit. There are two grids in that. Yeah. Yeah, one of them is held in a support, which we need when we are mounting on the microscope. Another one is a little open. But you will not be able to see how the squares look like. You need a magnification, you need some kind of microscope to look at that. So I just wanted to show you all, because I can't show you an electron microscope. When you actually image, it's very different from a light microscope. Like light microscope, you just turn on the light and then you can see what you want to see, right? So in an electron beam, because it's a very big microscope, right? Like it's almost like a 6 feet microscope, and you have like a narrow, tiny beam. So first we spend a lot of time, you'll see that we align the beam first. It has to be aligned into the optical axis. So there are some procedures that we do, but this is how a screen will look like. You're first trying to set the center, you're trying to make the beam circle. Sometimes it can become elongated because of the lenses that you're using. So you'll do all that, and then it takes a little bit of time to first just set the beam only. And then when you are imaging also, which I'll show you, so that is how you do alignment. Then when you are imaging also, like in light microscope also you will see, right? You first do lower magnification imaging. You see where your object of interest is, and then you go there and then change the higher magnification. So in electron microscope also you do that. You first see how your cell looks like, then you zoom into one particular area, and then you try to do that. So there will generally be different ways. This I'm not talking about this, but this is needed to set the height of the microscope, because height at which your image is. You are moving the screen, and then you go to your area of interest. So the scale here is in the nanometers, and also you have to move very carefully. It is not, you can't move the whole slide here and there. It's like a small, you saw that support, right? That is what you're moving. So what you're seeing, so I'm seeing in the nanometer scale. So that's how you take an image. So I'll show you actually some real images. So what happens is, this is a little complex, but I'll just explain it to you all. So if you have your sample, just imagine like what you're seeing, that grid when I mount it, the sample is in that holes. Okay, so it is suspended in the hole, and there is interaction of the electron from top. So you can imagine all these small, for example, here I'm just taking one protein molecule, one complex which is inside the cell. So when it interacts with the electron beam, what image you get is a projection. So all of that 3D information is collapsed into a 2D micrograph. We call it a micrograph. So this is the kind of image that you will see. And this has all kinds of views of your protein of interest. So whatever your complex is, you will have different views because it's all suspended, right? You have different kinds of that. So you will see all different orientations of your molecule of interest when you pass an electron beam. So a typical micrograph that you will get, you will see, so generally black and white is not as colorful as you see in light microscopy, but you have a lot of details, like what you want to see inside, that lots of details are there. So from this, you can actually identify where is your molecule or protein of interest. So whatever you're working with, how do you look at that? There are different views that are possible. So what is interesting in electron microscopy is, with this information, I can get 3D volume information. So I want to ask you all this question. If I have a 3D object, okay, I don't know how it looks like because it's very, very small. If I want to reconstruct that, what strategy will you use? Like, how will I reconstruct a 3D volume? So I think you all know light microscopy very well, so all of you have said slices, but okay. So slices is correct if it is big object. Now I'm talking about the size of this is 5 nanometers. No stage can slice at 5 nanometer. Yes, good. So that's the way. Can I just borrow your water bottle from me? So if this is your 3D object, okay. So what you all told is, one way I can make the 3D reconstruction is I take slices. Okay, so I take different slices, put them all together, you will get a 3D object. If this is very small, I don't have a slicer, I can't slice beyond microns. If it is so small, I can't focus, there's no microscope which will allow you to focus at the nanometer level. Another way of doing the 3D reconstruction is, I take this object and I put light in different directions. So I put it in this direction, you will get one image. I'll put it in this direction, you will get another image. I'll put it in this direction, get another image. Now all that images, if I know the angles, how they look like, I can put it back in 3D space and I can get back this object, right? So that is one way of doing it. So that is what is used in electron microscopy. So
Is there a chance we can use smaller light tubes that make light slices? Because there's one metal microscope which I read from the previous lecture now in your data.
Even with that, because of the physical limitation of light, the maximum information you can get is still in around 40-50 nanometers. So if you want to go below that, you can't do it with light. So you need to change to a different light illuminating source, which can be X-rays or electron beams. but with those you can do it. But with light also you can do this kind of method. With light it's easier to just do a Z-stack, what they say. So you just, because at that stage you can section at different levels and you can get that information. But with electron microscope, now you can't do that. So you do this, you do projection, and then you can do it's called a back projection, and then you can get the 3D volume from that. Do you all understand? So even if when I'm standing, if you take a picture of me from front, from side, from this side, from behind side, and I tell you this is how it is, you will be able to reconstruct a 3D image of me, right? How I look like, roughly. You can get this information. So that is what we do in electron microscopy. So you can put together all this information to get a 3D volume. So I'll show you one reconstruction example. So this one only. So you can do this for cells and you can also do this for proteins, right? So if I am removing a protein out of cell and taking an image, it will be oriented in different orientations. So when I have a pure protein in a test tube, right, and I put it on that small grid that I showed you all, it will be randomly oriented. So when I take any image, you will see random orientations of that. So that you can combine now. You can make classes. So we do this. We can make different classes of the orientations. I'm very briefly telling this. I can tell you all, like if anybody wants to know more, I can show you all more videos about this, right? Yeah. So you can make different orientations of this, and then you can project and get a 3D volume, right? And this is how a 3D volume looks like of this object only. So you can get all information of protein. So how it looks like, the interactions and all. And you should look at the scale. I'm in nanometers. So what you're looking at is very, very small, right? So I have some research examples. I'm thinking, do you all want a break? Like, is there a break at this time? Yeah, 5 minutes you can take a break. Okay. So after this, I'll show you some research examples. So I have some videos of what we do in research using electron microscope. Yeah. Okay. So I showed you this 3D volume, right? Like, so you can also look at, you can use electron microscopy to look at the whole cell. That's what I said in the first part, right? So you all know about Plasmodium falciparum, which causes malaria. Yeah. So you might have seen this kind of images where inside the RBC you'll see the parasite. So you get like small details about the parasite, right? Now, if you like a little bit about Plasmodium, it lives in both humans and in mosquitoes. So part of its life cycle is in the human body, like in RBC and in liver cells, and part of it is in the mosquito. And all of them have different stages. So you don't need to know this detail, but they are called by different names like sporozoid, merozoid, you have gametocyte, and then you have ookinete. And they all are different forms. This is the same parasite, but when it goes into different cells, it has different forms. So what we can do is, we can use this whole parasite and do electron microscopy on that. So you can see how the parasite looks like. So one stage I will show you. So this is like the sporozoid stage. So when you look at it in fluorescence microscopy, it will look like this. Okay. When we put it on the grid, it will look like that. The grid is what I showed you, no? That small thing. So that it will look like that. So if you can see these small parasites here, that is what the parasite is. And then I can make a 3D volume of it using what I showed you, no? I take different images and then back project and make a volume. And then you can see something like this. So now you can see all the details within the cell, how it looks like inside a cell, all the information you can get, right? And you can do it for different stages. Now so I did it for sporozoid stage, which is inside a mosquito. I can also do it for merozoid, which is inside your blood cells, like your RBCs. So here if you see inside light microscopy, you will see like this small, And then on the grid, the small ones that you can make out, those are these cells. And then you can have a 3D volume of that here. So we can identify from this the different features of the parasite. So I can identify that this is like a membrane, this is the nucleus, there are cytoskeleton like microtubules, which is my next topic actually. So you can do all this information and then you can study the parasite, like how it behaves, how it does. So this is one way of looking at this. Now I actually started this lecture with virus, right? So wanted to show you virus images. So this is the SARS-CoV-2. So the image that you saw in the beginning is from electron microscopy. So you can't see it with light microscopy. So scientists, when the pandemic was going on, scientists isolated the virus and they did electron microscopy. So this is how the image looks like. And after we do a 3D reconstruction, this virus, it looks like that. So I have a video to show you all how it looks like. So this is how, I hope it plays. Yeah, so that is a virus. So those are the different viruses that you see. So you can see all the details, right? What you couldn't see by light microscopy. And then you can, we call it segment out. You can identify how these are organized. So what you see on the top are these corona proteins, like, which help it to attach to our cells, right? And then you can get very high resolution information. So now what I'm showing you is overall information, but you can get information about each of this protein and all that also. So I think that time you would have heard it interacts with the ACE2 receptor, which is present in our cells. And then when there was new variants being produced, there were mutations which was happening, right? So that interaction was changing. So all that information you can get from this, like from getting a virus structure and how it looks like. So I can just show it to you all once again. And look at the scale, it's all in nanometers. So you are looking at something which is so small using an electron microscope. So each protein, you can get the detail of it, like what is happening. That much you can zoom in and get the information, right? Right, okay. So that is basically about electron microscopy. So how you can use electron microscopy to look inside a cell, see what is going on, right? So that was my first part actually.
Swayam Prabha, Digital India, Educated India.